Hello everyone, Lauren Bean here. This is a lecture for chapter 12, which covers the central nervous system. So we're going to be specifically talking about the brain and spinal cord for this chapter. For CNS development, it begins with a process called neurulation, so the formation of these nervous system structures in the brain. It all begins as a very simple neural tube. And over time, and quite quickly, following fertilization of egg and sperm, forming a new individual, we start to see these primary brain vesicles form from the neural tube. The primary brain vesicles, or these primary swellings, then give rise to secondary vesicles and eventually produce adult brain structures. You don't need to worry about memorizing all of the different vesicle types, all the terminology here. To keep it relatively basic, I just want you to understand the fact that it starts as a very simple neural tube, which gives rise to the brain in the spinal cord. For major regions of the CNS, these are the adult brain structures. So we have the cerebrum. This is this lightish tan color here. It's the largest structure, about 83% of the brain mass, and it's also the most superficial. When we compare our brain to other animals, it's really the cerebrum that has increased in size and it has become more complex, making us, intelligent and unique amongst other animals. These other brain structures are relatively the same when we compare humans to other animals. So we refer to that change as cephalization, an increase in the head size, an increase in the cerebrum. The diencephalon is shown in purple. This is involved with integration, bringing information together, relaying information to the correct area in the brain. Hypothalamus is in there, maintains homeostasis, controls autonomic functions. So we'll look specifically at the diencephalon and talk about those parts and pieces when we get there. Then we have the cerebellum, the little brain. This is a separate unit. It's separate from the cerebrum, and you need to be careful with terminology. Cerebrum superficial, cerebellum is posterior and inferior. It's a little brain in the back. It has all to do with motor coordination. Then in green, we have the brainstem. It includes the midbrain, pons, and medulla oblongata, collectively controlling very primitive autonomic functions, right? Controlling normal heart rate, breathing rate, all of that good stuff. Also serving as a connection point between the spinal cord and these bigger brain structures. The spinal cord itself is really just a big electrical cord that transmits information to and from the brain, and also is a major reflex center, which we'll talk more about reflexes in chapter 13. Brain lateralization refers to the fact that certain functions are more dominant in one hemisphere in comparison to the other. So when we look at the left brain functions, it's involved with analytical thought, sequential processing, so conducting something in kind of like a linear manner. Language, science and math, writing, number skills. So certain functions like that are carried out more on the left side of the brain. So you may hear people say like, oh, I'm left brain or I'm right brain. So some people are better at these left brain functions than they are right brain functions and vice versa, right? You might know someone or maybe yourself, you're really artistic, creative, imaginative, holistic. You love music, right? You're intuitive. So you may have more of a right brain dominance occurring within you. Of course, we have both of these properties, and there's always communication between the left and right hemispheres. But again, we may find that some individuals are a little bit more dominant on one side compared to the other. For the lobes of the cerebrum, we have the frontal lobe, parietal lobe, occipital lobe, and temporal lobe. We'll notice some important landmarks here. So the central sulcus divides the frontal and parietal lobes. And a sulcus refers to a shallow groove on the surface of the brain. To either side of the central sulcus, we have a pre-central gyrus and a post-central gyrus. A gyrus is a ridge, gyri for plural. This structure here is going to be really important when we start to look at the function of the cerebral cortex. It divides motor functions from sensory functions. So I'll reference back to these areas when we get to that point in the lecture. On the lateral aspect of the brain, we also have a lateral sulcus dividing the temporal lobe from the other lobes. And then we have the transverse cerebral fissure, much deeper groove, providing separation between the cerebrum and the cerebellum. There's actually no connection here at this fissure between the cerebrum and cerebellum. The only contact between these two structures is through the brainstem. So they're very separate and very isolated from each other. Another lobe that we couldn't see on the previous page is the insula or the insular lobe. It's buried underneath the temporal, parietal, and frontal lobes that has specialized areas that carry out certain functions that we'll see here in a few pages. Deeper in the cerebrum and deeper in the brain are the brain ventricles. These are fluid-filled chambers that contain cerebral spinal fluid. So we have a pair of lateral ventricles, kind of very horn-like shape to those. There's a third ventricle and a fourth ventricle. We have interventricular foramen connecting the lateral ventricles, draining fluid into the third, as well as a cerebral aqueduct between the third and fourth ventricle. So the fluid is continuously produced, continuously reabsorbed, and it flows through these structures. So the brain is not just one solid mass of tissue, it does have these fluid-filled chambers. I'll talk more about cerebrospinal fluid toward the end of the chapter, and we'll touch on these structures again and consider the flow. One additional thing I want to point out is septum pellucidum, which is a thin membrane that is separating the paired lateral ventricles. You can see the septum pellucidum when you do a brain dissection, and I'll point that out in the sheep brain dissection video. Looking at the cerebrum, what's very obvious is unique gray and white matter, so darker areas and lighter areas of the tissue. Gray matter consists of neuron cell bodies and dendrites, versus a white matter mainly consists of myelinated axons. It can contain unmyelinated axons, but just really kind of thinking about the myelinated ones because myelin is what gives that white color to the tissue. With this frontal section here, we can see the distribution of gray and white, and it creates these major regions of the cerebrum. So the cerebral cortex is this superficial layer of gray matter. Again, this is where the neuron cell bodies and dendrites are being found. Very, very important area that we'll talk more about. Deeper in the brain, we have basal nuclei or brain nuclei. These are islands of gray matter. These function as little groups. They process information. There's a site of synapse going on there, process motor, control, sensory processing. I'll briefly talk more about those as well. And then all of the white matter that you see just represents axons that are transmitting information to and from, to the brain nuclei or to the cerebral cortex, from this area of the cerebral cortex to that area, right? There's a ton of different axons that are connecting everything together. Most important thing I want you to understand from this is that neurons are highly organized in the brain. It's not just a random, mix of neurons everywhere. So when we look at the cerebral cortex, you can see kind of this overlay of neurons here. The cell bodies and dendrites are making that gray. The white are just these long axons. So again, the cerebral cortex is that superficial gray matter of the cerebral hemispheres. It's relatively thin, only about 2 to 4 millimeters. So it's all the cell bodies, dendrites, some glial cells, blood vessels, but no axons here in the gray matter. A lot of different important roles that are carried out here in the cortex. We're going to look at really specific areas um, when we get into it. We'll identify auditory areas, visual areas, so on and so forth. But here's kind of a cohesive set of functions carried out by the cerebral cortex. So sensory perception, motor control, cognitive functions. So that's more of our higher level of functioning, like thinking, problem solving, decision making. It processes language, both production and interpretation. It contains an area of regulating our emotions. It contains our personality traits, memory, and learning. So, a lot of real critical functions here being carried out by the cerebral cortex. There are three main functional areas in the cerebral cortex. So, you have sensory areas, association areas, and motor areas. All in red here represent motor areas controlling voluntary movement to some extent. It's found anterior to the central sulcus. In particular, we have this precentral gyrus here containing the primary motor cortex, kind of the starting point here of motor function. But all of this other red carries out motor activity as well. In blue represents the sensory areas, so that starts at the postcentral gyrus and includes other posterior regions shown in blue here. We're going to see with sensory areas, there's unique areas for the special senses, so vision, hearing, olfaction, gustation, that's smell and taste, respectively. And then there's also a lot of association areas, which integrate and interpret information. The pink on this image represents what we call multimodal association areas, very complex association areas such as the prefrontal cortex, posterior association area, and we will cover those areas after we go over motor and sensory regions. Starting with our motor regions at the primary somatic motor cortex, this is found at the precentral gyrus of the frontal lobe and is responsible for initiating voluntary movements to control fine motor skills. This area of the brain consists of pyramidal neurons. They're named according to the pyramid-shaped cell body. The axons extend to form pyramidal or corticospinal tracts that connect this primary motor cortex to the spinal cord. As they descend down the spinal cord, they're going to synapse with what we call lower motor neurons, which will then go and innervate the skeletal muscles to control that motor output. Okay, so very specific location, very specific function, very specific cell type making up the primary motor cortex. The primary motor neurons control voluntary movement on the opposite or contralateral side of the head and body. That's a very important statement. So contralateral, right? The left motor cortex controls the right side of the body. Right motor cortex controls the left side of the body. Things are contralateral because the neurons decussate or cross over in the medulla oblongata, right? So contralateral and decussation are two important terms you need to understand. This becomes especially relevant when we think of injury to the brain, stroke, lesion formation, neurodegenerative disorders, right? Because it's going to impact the contralateral side of the body. The neurons in the primary motor cortex are precisely organized according to the region that they control. So this forms what we call a motor homunculus, a map of the body on the primary motor cortex. So this image is very, very much specific. You know, this area of the primary cortex is controlling the muscles in the foot versus this area way over here is controlling muscles of the face. It is precisely mapped out. Each neuron innervates a particular area in the body. You'll also notice that certain body parts are larger than others. They seem like exaggerated. And that is specific to the level of innervation or the space occupied on the primary motor cortex. So these larger body parts, it's implying that there's more precise control of those muscles. We have much more precise control of our hand and our fingers opposed to our elbow and arm and body trunk, right? So the distortion is for a reason. The premotor cortex is the preparatory area, right? It's pre-motor. So it functions to help plan and organize movements. It integrates sensory information to guide our movements, right? So we need kind of that sensory input to dictate motor output. So that integration, that coming together of sensory and motor processing takes place there in the premotor cortex. prepares the body for movement, and also plays a significant role in learning and adapting to new motor skills since it can take in that sensory guidance. Unique motor areas include the Broca's area and the frontal eye field. The Broca's area is the motor speech area controlling muscles of speech production. So I always think I don't want my motor broke. Motor speech area. For about 90% of individuals, the Broca's area is found only on the left hemisphere. Call it the dominant hemisphere because of that correlation of about 90% of people having the Broca's in the left. And along with that, individuals write with their right hand. So there is that correlation with language and language expression, all due to that contralateral establishment in the brain. The corresponding region in the non-dominant hemisphere is responsible for expressive nonverbal communication, hand gestures, facial expressions. So this corresponding area, I'm saying if the Broca's is on the left, the corresponding non-dominant region would be on the right. Still contributing to language to some extent, but it's more that motor expression for nonverbal communication. And then the frontal eye field controls voluntary eye movements. The extraocular eye muscles are the most precisely controlled muscles in the whole body. So it makes sense there's a whole frontal eye field dedicated to its control. Moving on to the sensory area, we have the primary somatosensory cortex. Primary telling us this is the first place sensory information is going, located at the post-central gyrus, so just posterior to the central sulcus. And the function is sensory perception. So the retrieval of pain, temperature, pressure, touch, vibration, as well as proprioception, which is position sense, an understanding of our body's position in space. The information comes from muscles, tendons, joints, telling the brain, what our body is doing. All of that sensory input first goes to that primary somatosensory cortex. So it receives information from many different receptors out in the body. We'll talk about those sensory receptors in chapter 13. So we find a somatosensory homunculus very much like we do a motor homunculus. Sensory nerves project to very specific locations on the postcentral gyrus, and that creates this somatosensory homunculus. Right, this has been figured out by neuroscientists, right? Know exactly where the sensory nerves are projecting onto the brain. This really allows for spatial discrimination being organized in this manner. Spatial discrimination, knowing, exactly where a sensory stimulus is being applied, exactly where a mosquito has just bit you, right? Very sensitive areas occupy a large amount of the sensory cortex. So our hand, our fingers, the sole of our foot, genitals, the face, internal organs, the tongue, right? Those are quite large here because they're really sensitive. Versus the head, neck, trunk, hips, legs, knee. I mean, they are sensitive, but not super sensitive in comparison to these other areas of our body. The somatosensory association cortex makes associations with that sensory stimuli. So it integrates sensory inputs, creates a comprehensive understanding of objects. It could be things like size, texture, how much space something occupies. It enables stereognosis, the ability to recognize something by touch. It integrates sensory experiences to really make sense of the sensory stimuli. And this association area is quite broad, quite complex. A lot of different connections occur in the cerebral cortex for association and interpretation. Specialized areas are for vision, hearing, smell, taste. So first here for vision, we have a primary visual cortex and a visual association area. Vision is processed in the occipital lobe, the back of the brain. So make sure you make that connection of vision with occipital lobe. Primary is where the vision or the visual stimuli first goes, then it's connecting to association areas for interpretation. We'll talk more about our special senses in chapter 15. So now you just want to think about the fact that there's two areas here, and that's concentrated in the occipital lobe. Auditory areas, there's a primary and an association area. This occurs in the temporal lobe. So primary is where sound information is first received. Association is where it is interpreted. Close to that, we have the Wernicke's area. So the Wernicke's area is more of a complex structure. We include it as a multimodal association area just because it is so complex, but it's responsible for interpreting spoken language. So it does play that auditory role, but also interpretation of written language and even sign language. So all of that language processing occurs in the Wernicke's area. This is also typically located on the left hemisphere, what we call the dominant hemisphere. Same idea as with the Broca's area here, okay, typically just on the left side. The olfactory cortex, olfaction refers to our sense of smell. So the primary olfactory cortex, it's responsible for perceiving odors. This type of sense, special sense, this cortex area in particular is unique because it is directly connected to our limbic system, our emotional brain system. And this explains why smells can really trigger a powerful emotional response, or even can trigger a vivid memory, create nostalgia, right? Create comfort, or create, repulsion, disgust, anger, fear. Sense of smell has a very strong emotional connection in the brain. Anosmia here refers to the loss of the sense of smell. That can be due to damage in this particular area. This can be a real safety issue for individuals. You lose the ability to smell smoke, inability to smell a gas leak. If you think of other animals, inhibit the ability to smell predators. So having an adequate sense of smell is very important. There are additional sensory areas located in the insula or the insular lobe. So we have the vestibular cortex. Whenever you see the word vestibular, vestibule, vestibular apparatus, always think about equilibrium and our body's balance. So this area processes sensory input coming from multiple areas so we can have a conscious awareness of our balance and position of head in space. So it receives input from the vestibule in the inner ear. So that includes like our semicircular canal, saccule, utricle. We'll get to that in chapter 15. It includes other proprioceptors out in our muscles, tendons, and joints. So we have a very good understanding of our body's position in space. The gustatory or taste cortex, gustation refers to a sense of taste that's also found in the insula, as well as the visceral sensory area as well. Visceral organs are our internal organs, like our stomach, our intestines, our heart, our liver, all of that stuff. This allows for conscious perception of sensations coming from those organs. Hunger, fullness, a full bladder, right? When we need to go pee, your lungs burning from exercise, so on and so forth. Then in pink, we have multimodal association areas. Multimodal meaning it involves several modes. It also involves several lobes. So it's a mixed bag of different things. It allows us to have meaning or apply meaning to sensory information, form meaningful actions, control a lot of more cognitive, higher level processing and functioning, right? So this is really controlling like conscious things that we are aware of, and these areas make us who we are, determine our personality, right? And our emotions. One specific area is the prefrontal cortex or the anterior association area. This is our most complex cerebral cortex area. This is our thinking brain. This is what makes us who we are. It depends on feedback from our social environment, from our upbringing. Again, the prefrontal cortex is really what determines who we are as individuals. It processes cognition or reasoning. It dictates our intelligence, our intellect, recall, personality. It is involved with our working memory, the ability to hold on information to carry out a task, complete an activity. It controls our personality expression as well as our social behavior. So, a lot of really complex things going on here in the prefrontal cortex. The posterior association area, this involves the temporal, parietal, and occipital lobes. So it helps to integrate sensory information from those areas. So visual, auditory, and other somatosensory information brings it all together, right? This is an association area, right? So it connects it all. It's important for spatial awareness, important for perception, recognition of patterns, recognition of faces, right? And putting it into context, knowing if someone is significant or making it relevant to your past histories. And the Wernicke's area is a major area in there as well, involved in understanding written and spoken language. That's a very complex process, which is found within this posterior association area. Another multimodal area is our limbic association area, which is a part of the limbic system, which as a whole really controls our emotions and behavior, attitude, kind of all those things. So the association area in particular just really integrates and associates sensory information and provides emotional impact, makes things either important or not important to us. So it includes the cingulate gyrus here, found just above the corpus callosum. It involves the parahippocampal gyrus down here as well. So these really internal areas of the cerebral cortex, which are all really intertwined with the limbic system. Now transitioning to talk about the brain nuclei, what we call the basal nuclei, these islands of gray matter deep in the brain. It includes several different specific areas. There's caudate nucleus, globus pallidus, putamen, subthalamic nuclei, substantia nigra, nucleus accumbens. A lot of different unique areas deep in the brain, and each region providing a specific function, but the overall thing that they provide for the body is the regulation of voluntary motor movement and motor control. And even more importantly, the suppression or inhibition of undesired movements. And that produces really smooth and coordinated muscle activities. An easy way to understand the importance of brain nuclei is to consider what happens when there's dysfunction in these areas. So, diseases such as Parkinson's, Huntington's, Tourette's, even OCD, these are all conditions when there is dysfunction in the brain nuclei. For Parkinson's disease in particular, it's due to the degeneration of dopamine producing neurons in the substantia nigra, one important area of the brain nuclei. And this causes individuals to have tremors. They shake a lot. They'll have rigidity, so stiffness in the limbs, stiffness in the body, very slow movements. They have issues with posture and with balance. So it's the tremors in particular that becomes a very significant symptom. And it really illustrates how, if there's dysfunction in the brain nuclei, an individual lacks the ability to have smooth, coordinated motor outputs. That suppressive property is not occurring appropriately. Next, we're going to consider cerebral white matter. White matter consists of myelinated axons shuttling information to and from, and it's made up of three fiber types: association fibers, commissural fibers, and projection fibers. Association fibers connect different regions within the same hemisphere, and this is important for integration. So this is when you have a primary area being connected to an association area. The association fibers are what are transmitting and connecting those areas in the cerebral cortex. Commissure fibers are connecting the left and right cerebral hemispheres. Corpus callosum is the largest and most prominent commissure fiber. There are other types, but that's usually the main one that is discussed. And then projection fibers connect the cerebral cortex to lower brain regions. So to maybe the basal nuclei or to the brainstem and down to the spinal cord. For the projection fibers, we'll look at two unique regions, the corona radiata and the internal capsule. With this visual here, we just have a different view of those fibers. So red are the association fibers connecting different regions of the cerebral cortex in the same hemisphere. Here's our corpus callosum connecting left and right. And then here are our projection fibers going from the cerebral cortex and down to lower brain areas. So we have this thickened area on both side here that condenses to form an internal capsule. And this is just a dense bundle of projection fibers serving as a major pathway for both descending and ascending tracks. Descending are going to be carrying motor information, ascending are carrying sensory information. The internal capsule forms from these radiating fibers, or what we call the corona radiata, a fan-like network of projection fibers connecting the cerebral cortex down to this internal capsule. All right, so that's how the information would flow if we're thinking about descending, is corona radiata to the internal capsule, navigating down, maybe decussating in the medulla oblongata, right? And then those fibers continue on as tracks down the spinal cord. Moving away from the cerebrum, we're going to talk about the diencephalon shown in purple. This includes the thalamus, this egg-shaped structure, the hypothalamus, as well as the epithalamus, which includes the pineal gland and posterior commissure. The thalamus is described as a bilateral egg-shaped structure, so has two prominent egg-like bulges, and it contains many, many brain nuclei within, clusters of neuron cell bodies and dendrites. It's a site of synapse between different neurons. And the thalamus is called the gateway to the cerebral cortex, or the relay station to the cerebral cortex, because all sensory information coming up the spinal cord into the diencephalon, it's going to stop at the thalamus. Thalamus is going to sort, relay, edit, synapse, and then send that information to the appropriate region of the cerebral cortex, depending upon what that information is. Is it coming from a touch receptor, pain receptor, right? Light receptor, whatever. The thalamus is what relays that information. So it's really the gateway to the cerebral cortex. With the hypothalamus, this is such an important brain structure. It's quite funnel shaped. It's below the thalamus, that's why it's hypothalamus. And there are many, many nuclei packaged within this area. For function, so many critical things here. Controls the autonomic nervous system, which maintains general body homeostasis. It interacts with our limbic system, so it influences our emotions. It regulates sexual behaviors, regulates our reproductive biological clocks. So, particularly for females, really regulating menstrual cycle, regulates the endocrine system, which has all to do with producing hormones. The hypothalamus regulates the endocrine system by controlling the pituitary gland, which hangs from it here. The hypothalamus makes hormones, which can influence the pituitary gland to make hormones, which can then stimulate other endocrine organs in the body to make their own hormones. But it's the hypothalamus that's the major regulator of the entire endocrine system. Going along with maintaining homeostasis and autonomic functions, it maintains our body temperature, or it's our body's thermostat. regulates food intake, water balance, so it can stimulate thirst, controls our sleep and wake cycles, right? It can influence melatonin release, circadian rhythms, seasonal changes. So many real critical functions here carried out by the hypothalamus. The mammillary bodies are paired, kind of pea-like shaped nuclei that bulge from the hypothalamus. They are part of our limbic system, that emotional system that we have. They're involved with processing episodic memories, so personal experiences and the memories that they produce in kind of an episode-like or in an episodic manner. They contribute to spatial memory, the ability to navigate, recognition of familiar places, objects, people, really helps to integrate olfaction with memory and emotional responses as well. So, very unique little bodies here that have quite complex function. For the epithalamus, this includes the pineal gland, which secretes melatonin that has a sleep inducing effect. And it also involves the posterior commissure. So, these are commissure fibers, axons that are going to be connecting left and right cerebral hemispheres. This area in particular is involved with the pupillary light reflex, so it controls constriction of the pupil in response to light, and it controls other visual reflexes like vertical gaze, synchronizing and stabilizing our eye movements as well. And it makes sense in terms of location. It's more posterior. Vision is controlled in the occipital lobe, kind of bringing those into proximity with each other. For the brainstem, this includes the midbrain, pons, and medulla oblongata. It connects the brain and spinal cord together, and it functions to control very primitive, programmed, automatic behaviors. So it sets a breathing rate, heart rate, blood pressure, all of that automatic stuff that we don't think about. Our brainstem structures are very similar when we compare it to other animals, right? So these structures and these functions evolved very early on. Another unique thing is that this is really the origination point for many of our cranial nerves, which innervate structures of the head and the neck, which we'll see in more detail in the next chapter. We'll start by looking at the midbrain and consider important features. The cerebral peduncles, these are these bulges here on the ventral surface of the midbrain. They bulge like this because they contain the corticospinal tracts or the pyramidal tracts. So those neurons originating from the primary motor cortex, traveling down the brainstem to get into the spinal cord, synapse with other motor neurons, which then innervate the skeletal muscles. So it's really a relay area for motor commands. Then looking at this cross section, we find some other distinct areas. Substantia nigra, this brown area here. Nigra means black. It's really more like a brown color. This contains neurons that produce dopamine, which is really important for regulating movement. It's important for reward pathways, has a lot to do with motor control. Parkinson's disease is the deterioration of the substantia nigra area. The red nuclei, very red in appearance, has a high level of iron containing molecules, giving that coloration. This also contributes to facilitating fine motor control, controlling limb movements, so contributing to that whole motor control concept. Then we have the periaqueductal gray matter. So that is surrounding the cerebral aqueduct. That is the tube that transports cerebrospinal fluid from the third to the fourth ventricle. The neurons in this area play a key role in pain modulation, so kind of regulating pain pathways, defensive behaviors, and also fight or flight response. On the dorsal or posterior aspect of the midbrain, collectively it's called the tectum. A unique area is the corpus quadrigemini, which collectively refers to these four bulges here. There's a pair of superior colliculi, a pair of inferior colliculi. For the superior, it's involved with visual reflexes, so tracking moving objects, coordinating eye movements. That's kind of just an automatic thing that we do, right? We catch something in the corner of our eye, we turn toward it, and we follow it. Similar with the inferior colliculus, but these contains auditory reflex centers. So involved with even like a startle response, right? We hear a loud noise, we turn toward it, we want to try to identify where that sound came from. So superior visual reflexes, inferior auditory reflexes. With the pons, the middle region here of the brainstem, this is the bridge or connection point between those two structures. So it's really just a big communication hub. It also is significant for providing communication between the cerebellum and the cerebral cortex. So neurons have to navigate through the pons to connect those two areas. When we look at a cross section of the pons, the fourth ventricle is found and it separates the pons from the cerebellum. We have some cranial nerves originating from the pons, and it plays a significant role in something called reticular formation, which we'll talk about, that regulates our state of wakefulness and arousal, and sending sensory information up to the brain. Lastly, the pons plays an important role with the transitions between our sleep stages, even switching into REM sleep as well. We're not going to talk a lot about sleep stages in this chapter. It's a pretty complex thing to discuss, and for time's sake, I'm going to kind of skip that conversation. For the medulla oblongata, this is most distal part of the brainstem, blends into the spinal cord at the foramen magnum, and it plays a crucial role in autonomic reflexes. One of its most insignificant or important things is it has cardiovascular and respiratory centers. For the cardiovascular centers, we can identify a cardiac and a vasomotor center. The cardiac center adjusts the force and rate of heart contraction. So at rest, it's going to establish a nice normal heart rate, normal force of contraction. It can be altered depending if we're in a sympathetic state or not. Then the vasomotor center is going to regulate or adjust blood vessel diameter, which in turn impacts blood pressure. Then for respiratory centers, it generates a rhythmic respiratory rate, the rate of breaths, but also the depth of breathing as well. And that is very dependent upon the carbon dioxide levels in the blood. That's really what influences our respiratory rate and the depth, right? Are we at rest and are we ventilating normally, or are we exercising and we need to increase that rate of respiration? For the respiratory centers, the medulla and the pons work together to control that. Then there are many other regulatory centers, reflex centers, which control vomiting, hiccuping, swallowing, coughing, sneezing. A lot of that is carried out in the medulla oblongata. Important structural feature of the medulla are the pyramids on the ventral surface. These are bulges that the pyramidal corticospinal tracts form. These are really big tracts. There's a lot of axons, a lot of innervation down to the skeletal muscles. So these pyramidal tracts form the pyramids, the bulges, as they traverse down the medulla oblongata. It's also the point of decussation where the tracks cross over. So that's a very significant thing to associate with our medulla oblongata. For the cerebellum, this is known as our little brain. It has all to do with motor coordination, ensures smooth, coordinated, voluntary movements. It develops procedural learning, or what we call muscle memory. The ability to just hop on a bike and ride it, even though it's been, 5, 10 years, whatever. Just that automatic muscle memory, that information is stored in the cerebellum based on neural circuitry. It controls timing and rhythmicity of movements. It can improve precision with time. It can correct muscle errors. It maintains our balance and our posture. So it receives a ton of information from all kinds of different sensory receptors and other brain structures, and it interprets, it processes all that information for adequate motor control. Structurally, there are unique features of the cerebellum. There are different lobes. There's two distinct hemispheres. The vermis is kind of the worm-like region in the middle that separates those hemispheres. It is heavily folded, forms what we call folia, that allows for compaction of a lot of tissue in a small area. There's a unique distribution of gray and white matter. So there's cerebellar cortex with gray. Arbor vitae is a term we use in reference to the white matter. Arbor vitae means tree of life. So it looks like a very elaborate tree when you look at the tissue and you kind of expand it. So again, arbor vitae refers to the white matter of the cerebellum. There are the cerebellar peduncles, which connect the cerebellum to the brainstem. So there's superior, middle, and inferior. Each of those specific peduncle areas provides a contact point. So superior cerebellum to the midbrain, middle cerebellum to the pons, inferior cerebellum to the medulla. So the peduncles provide that structural point of connection, also a navigation pathway for axons shuttling information. Most of the fibers to the cerebellum are ipsilateral, meaning they originate from and travel to the same side of the body. So there's no crossing over for the most part that occurs from neurons originating in the cerebellum. The cerebellum consists mostly of what we call Purkinje cells. These are very large inhibitory neurons found in the cerebellar cortex. Highly organized in that tissue. Here's a really cool image of that there. This is what one cell looks like. Kind of a flask-shaped cell body here. Very extensively branched dendrites. And then one single long axon. So these neurons receive a ridiculous amount of information and then carry out one motor output. And Purkinje cells release the neurotransmitter GABA, which is the major inhibitory neurotransmitter in the central nervous system, or in the brain in particular. So inhibition is just as important as excitation, and this inhibitory function enables these cells to really fine-tune the motor movements and prevent excessive or unwanted motor output. So an inhibition is very important in the cerebellum. This image here highlights the major regions of the brain, labels all the different structures. Most of these we've already covered, so I'm not going to go through every single component, but it is important that you can label this image for your lab quiz, that you can have a visual understanding of where these structures are found in the brain. Now we're going to consider two key functional systems in the brain, so the limbic system and the reticular activating system. For the limbic system, this has to do with regulating our emotions, behavior, motivation, memory, even olfaction, right? Our sense of smell is directly connected to the limbic system. We say it's a functional system because it includes many different structures kind of widely dispersed throughout the brain, a little bit more challenging to pinpoint it to one particular area. This image here does highlight all of the different components that contribute to it. The ones that we're really going to focus on are the amygdala, the hippocampus, but there are many other members, but I kind of got to draw the line somewhere. The overall function here is really to provide emotional richness to our life. So it integrates emotions with higher brain functions, which influence decision making, reasoning, right? All of those components of life. The amygdala is a significant part of our limbic system. It processes fear, anger, assesses threats to the body, If we use our example down here, like some people are terrified of spiders and they see a spider, that sensory information coming in, it's going to go to different areas in the body, visual thalamus, visual cortex, but will also be shuttled to the amygdala and cause that fearful response. Causes a physiological response, increases heart rate, breathing rate, blood pressure. Same things happen when we're angry or we feel threatened. So the amygdala is really what regulates that whole idea. With the hippocampus, this is involved with creating new long term memories and learning, really, in general. The name hippocampus comes from the scientific name seahorse because when anatomists removed it from the brain, it looked just like a seahorse, as you can see here. So it kind of has that hooked, weird shape. The hippocampus is really unique because it's one of the two places in the adult brain in which new neurons can be generated throughout life. So that new generation of neurons contributes to forming new long-term memories, right? And this enables us to always learn more, right? To always gain more knowledge for our entire life. So something really unique kind of going on there in the hippocampus. Through research, it's been discovered that animals who are exposed to extreme stress, it causes the hippocampus to shrink in size. It makes learning become really, really challenging. So, what people are dealing with in their emotional life, how, their upbringing has been, their social interactions, any stress applied to them can directly impact their ability to learn. In contrast, animals that exercise and continuously perform challenging tasks, they can increase the size of the hippocampus. So exercise is really critical for so many things in the body, but it enhances learning as well. When we talk about memory, there's different types of memory. We can identify short term versus long term. So there's a lot of a classification that can go on. Memory can be declarative, so memory of facts, names, faces, words, dates. I have a harder time with my declarative memory. I have a hard time remembering the name of people. Dates, oh my gosh, no way. I have a challenging time with declarative things. Procedural memory, that's more carrying out a procedure, more long-term skills, playing a piano, doing a particular type of memorized activity. Motor memory kind of tying in with that, memory of motor skills such as riding a bike, and then emotional memory as well. So specific things tied in with the limbic system. Memory consolidation is the process of converting short-term memories into long-term memories. Short-term memory, or what we call working memory, is when we just hold on to bits of information temporarily and then we forget about it. It's limited to about 7 plus or minus 2 pieces of information. So a classic example is like when we look up a phone number and we, temporarily remember it, dial it, and then make the phone call, right? We probably are going to forget that phone number in, a couple of minutes, right? It's just short-term working memory that allows us to carry out a particular task. Long-term memory, this is when, we remember it long term. This has really limitless capacity because we do have regeneration of neurons occurring in the hippocampus. There are significant factors that will allow for this memory consolidation or transfer from short term to long term. So, our emotional state, rehearsal, association, and just kind of an automatic subconscious level of establishment, right? These things are what really make us form long term memories. So, say, initially you look up a phone number and it's short term, right? But if there's a tremendous amount of rehearsal or repetition, you make that phone call once every single week, well, over time, you're going to remember it, right? And it becomes a long-term memory. Or I remember my childhood phone number that was in my childhood home. I'll never forget it because it was rehearsed. Emotional state, alertness, motivation, that strongly influences long-term memory. Like, I love anatomy and physiology. It's enabled me to remember the content because I'm alert, I'm motivated, I'm interested. Associations are really important as well. That's a very important component of learning. We need to make associations, we need to connect things together, right? To be able to remember them over time. And then some things just happen automatically, subconsciously, for some reason, right? We generate these core memories that just feel totally irrelevant and random. So it's kind of a mixture of different things influencing that transfer of short term to long term. Another functional system is the reticular activating system, or RAS. And this refers to a network of neurons that are receiving input from many different sensory systems and sending that information up to the thalamus and to the cerebral cortex. So it really plays a role in alertness, keeps us awake, conscious, aroused, but also is really important for filtering out repetitive or weak stimuli. There's a major filtration system at that transition point of sensory up to the conscious level in the cerebral cortex. It's believed that the RAS disregards or filters out around 99% of sensory stimuli in our environment. If it didn't do this, everything would drive us crazy. So it's a very important filter system that occurs, right? But it keeps us aroused and alert. It can be inhibited by sleep centers in the hypothalamus, right? Inhibited by melatonin, cause us to go to sleep. Depressed by alcohol, drugs, tranquilizers, it shuts this RAS system down. If there's injury to the reticular activating system, it causes people to be in a coma, right? This is a system that is impacted. And there are dysfunctions related to ADH and as well as insomnia, the inability to fall asleep normally. Here's a visual for you for the reticular formation or reticular activating system. So it's showing here the stream of sensory information coming up to the thalamus, stream of information going to the cerebral cortex. There's always this input of information going through this neural circuitry of the reticular activation. And this is what keeps the cerebrum aroused and alert, but there's also that filtration that's taking place within as well. Now moving on to talk about the coverings of the CNS, or what we call the meninges, provide several functions, mostly a protective structural role, protects blood vessel, encloses venous sinuses, contains cerebral spinal fluid, forms partitions in the skull, and from superficial to deep, it includes the dura mater, arachnoid mater, and the pia mater. When someone has a meningitis, there's inflammation or infection of the meninges. Can be very dangerous. It could lead to encephalitis. It could spread to the brain tissue, which is a very serious problem. Dura mater means tough mother. This is the strongest layer, strongest meninx. It's really composed of two layers, a periosteal layer, which is really just periosteum of bone, and then a meningeal layer. These two areas separate in certain regions of the brain and form dural venous sinuses. So here is labeling the superior sagittal sinus. So there's other dural venous sinuses. This is an area that collects venous blood from the brain. So deoxygenated blood contains a higher level of waste as well. It collects in the venous sinus and then eventually drains through the internal jugular veins of the neck, returning to the heart so it can be pumped to the lungs for oxygenation. So this is kind of like waste blood being drained and recycled. The dura mater also forms dural septa, inward projections that divide the cranial cavity, help to stabilize the brain, limit excess movement of the brain. So we have the false cerebri, we have the false cerebelli here, and then the tentorium cerebelli as well. So with the false cerebri, we see that it's dipping into the longitudinal fissure, kind of providing that compartmentalization of left and right. It's anchored to the crista galli of the ethmoid bone, an important attachment site to that cranial bone there. False cerebelli, that runs along the vermis of the cerebellum, dividing the left and right cerebellar hemispheres. Tentorium cerebelli, that is a horizontal fold over the cerebellum, extending into that transverse cerebral fissure, separating the cerebrum and the cerebellum. So again, overall function here really to limit excess movement of the brain, really stabilizes it within the cranium. Next layer is the arachnoid matter. So it's shown in this kind of transparent layer with all these weird little fibrous extensions. We have two spaces that are formed with the arachnoid matter. The subdural space is between the dura mater and the arachnoid matter. And then the subarachnoid space is below the arachnoid matter, and it contains cerebrospinal fluid. So that space here with all those weird little projections is the subarachnoid space containing the cerebrospinal fluid. Also contains large vessels in there as well. We'll see that there's areas in which the arachnoid matter project up through the dura. Call these arachnoid villi or granulations. This permits the reabsorption of cerebrospinal fluid, which drains into the dural venous sinuses. So cerebrospinal fluid is constantly synthesized and produced and constantly being reabsorbed and drained by the venous system. Then our last layer is the pia mater, found right on the surface of the brain, clings really tightly, following every single gyrus and sulci, every convolution of the brain surface. Cerebrospinal fluid's composition is mostly water, about 99%. It has standard electrolytes, sodium, potassium, calcium, all of that good stuff. Has low levels of protein in comparison to, say, the fluid part of our blood. Has glucose for energy, very, very few cells, and is a little bit acidic in its pH. Each day, the brain produces about 500 milliliters, but at any given time, there's only about 100 to 150 milliliters found circulating in the brain. So again, it's constantly being produced and constantly being reabsorbed and drained into the blood. For functions, it provides a liquid cushion. It helps to maintain an adequate chemical environment for the brain. Most importantly, it gives buoyancy and reduces the weight of the brain by about 97%, allowing it to kind of float within the cranium. Without it, the brain would be crushed by its own weight. And that's because brain tissue, it's heavy, but yet it's really, really soft, and it would not be able to tolerate that compression against the bone. Choroid plexuses are clusters of capillaries found within the ventricles of the brain, and they're lined with ependymal cells. So collectively, it's the choroid plexuses and the ependymal cells that produce cerebrospinal fluid. Then the cilia of the ependymal cells help to sweep and move the fluid throughout the spaces. With this image here, we're considering the flow of cerebrospinal fluid throughout the brain and the spinal cord. So the production of it begins at these red structures here, the choroid plexuses. So we see fluid is going to fill the lateral ventricle, go down the interventricular foramen, fill the third ventricle, down the slippery slope or the cerebral aqueduct to the fourth ventricle. Fluid can leave through lateral and median apertures, bathing the surface of the brain, being within that subarachnoid space. The fluid bathes the surface of the spinal cord and also travels down the central canal of the cord as well. When the fluid is at the surface of the brain, it can be reabsorbed by arachnoid villi or granulations. So these are projections of the arachnoid matter into these dural venous sinuses. That's where they're reabsorbed into the venous blood and recycled throughout the body. With the blood-brain barrier, we're focusing on the barrier found around blood capillaries that are in the brain. And a blood capillary are the tiniest blood vessels that we have in the body that are responsible for exchanging nutrients and waste, oxygen, and carbon dioxide. With this image below, we're comparing a standard capillary here to a brain capillary. And there's really two important features that we find in the brain capillaries providing that true protective barrier. So, one being tight junctions between the endothelial cells forming the wall of the capillary. And remember that the tight junctions are specialized cellular junctions. This really fuses those cells together, ensuring that things will not slip between the cells. And then the next layer of protection are these astrocyte phi here that wrap around the blood capillary. So here's our astrocyte in the background is extending its cellular processes. Here we have these flattened kind of bulbous feet wrapping right around. So it's the tight junctions in the astrocytes really forming the blood-brain barrier. So only certain molecules can pass through the capillaries and get to the brain tissue. This really helps to maintain a stable chemical environment. It prevents certain molecules and pathogens from infecting the brain. So it's a very, very important protective layer here. Now we're transitioning to talk about the spinal cord. An overall function here is to provide two-way communication to and from the brain and the body. We're going to talk more about the spinal cord in the following chapter when we dive into the peripheral nervous system, because it's going to be really relevant there, because it's the spinal cord really serving as the transition structure between the CNS and the PNS. But technically, spinal cord is part of our central nervous system. It contains a lot of tracts, shuttling information up and down. Another key thing is that it's a major reflex center. It initiates and completes reflex arcs, that we call them, for automatic, quick activities, which I'll talk more about in the following chapter. The cord is enclosed within the vertebral column, so it's the vertebrae that provide protection for the cord. It begins at the foramen magnum of the skull, and the true wrapped cord ends at about level L1, L2, and it terminates at the point called a conus medullaris, or medullary cone. From there, nerves just kind of radiate out and form the cauda equina, or the horse's tail. So there's a difference in structure here at that point. Along the cord, we also find these enlargements. So there's a cervical enlargement, lumbar enlargement. So this is just areas where the cord is a little bit larger because there's going to be a lot of nerves coming off of the cord at that point to innervate the arms and the legs. And then lastly, in green here, we have the filum terminale, fibrous extension of our medullary cone or conus medullaris that is anchoring the spinal cord to the coccyx or the tailbone. So more of a structural anchoring point. With a cross section of the spinal cord, we can see where the gray and the white matter is found. The gray matter is found deep, forming the core of the spinal cord, and then the white matter is more superficial. So it's the opposite of what we find in the brain. The gray and white matter is still composed of the same things. So gray matter is consisting of neuron cell bodies, dendrites. We see that it forms a unique H-like shape here, or a butterfly, and each of those extensions we call horns. So there's a pair of dorsal horns, a pair of ventral horns, and there's also some lateral horns, or really just kind of this side lateral aspect, not quite as obvious. The cord, as well as the brain, is really divided sensory and motor. So what we find here is that motor is ventral or anterior, sensory is dorsal or posterior. So the dorsal horns here contain sensory afferent neurons. The lateral horns contain cell bodies of autonomic neurons. Those are going to innervate our visceral organs. And then the ventral horns here contain the cell bodies of motor neurons that innervate our skeletal muscle. So it's very organized within the cord. You can see I put this line here, sensory and motor. The white matter consists of tracts, bundles of myelinated axons transmitting information up and down the cord. So there's an anterior region, posterior, lateral, and we're going to find that when we cover the different types of tracts that they are organized within the cord. We can describe them as either ascending or descending. So sensory tracts are ascending, going up to the brain. Descending are going down, right, carrying motor information. As those fibers leave the spinal cord, they're going to form roots. So there's a ventral root and there's a dorsal root. The ventral root's carrying motor axons, the dorsal root carrying sensory axons, right? It's still keeping that same type of organization. On the dorsal side, what's unique is there's a dorsal root ganglion, and this is a collection of neuron cell bodies, particularly sensory neuron cell bodies. Following that, we see that the dorsal and the ventral root come together to form a nerve, what we refer to as spinal nerves. And these are mixed because they contain both dorsal and ventral roots, sensory and motor axons. We'll dive into nerves in more detail in our following chapter. With this visual here, we can see where the specific ascending and descending tracts are found. We find that most of the ascending sensory tracts are concentrated in the dorsal region. So these two blue areas are the dorsal white columns. And then a bulk of the descending motor tracts are found in the ventral region. You will see, that they are distributed throughout. It's not a perfect division of ventral and dorsal, but the overall trend is that sensory is dorsal, motor is ventral. Then considering neuron pathways, how the information is transmitted either up or down. With ascending sensory tracts, most of the time there's 3 neurons involved in that pathway. So we call them first, second, and third order neurons. The sensory receptor, the sensory neuron, is considered the first order neuron. It's the one detecting a sensation and then transmitting that information to the spinal cord or the brainstem, depending upon, where we're finding that sensory neuron. So it's going to synapse with a second order neuron, which will then take that information to the thalamus. 9 times out of 10, the information is going to go to the thalamus, not always. So this is kind of a representation of a general sensory pathway. So the second order neuron is going to synapse with a third order neuron in the thalamus. That third then takes it to the appropriate area of the cerebral cortex, right? It's going to take it to that appropriate somatosensory map, depending upon where that sensation came from. Versus with our descending motor tracts, there's typically 2 neurons involved in the pathway. Call it an upper motor neuron and a lower motor neuron. The upper motor neuron originates in the cerebral cortex, particularly in the primary motor cortex. We're talking about pyramidal cells most of the time here. That neuron is going to travel from the brain down to the spinal cord, maybe the brainstem, depending upon what that motor neuron is, synapse with a lower motor neuron, which will then innervate an effector cell, innervate a muscle, innervate a gland, and carry out that effect. There are 4 major ascending sensory tracts in the spinal cord. There's more than this, you can get much more specific, but to keep it pretty brief, we're going to really just refer to these 4 major ones. So we have the dorsal columns, we have lateral spinal thalamic tracts, anterior spinal thalamic tracts, and spinocerebellar. Notice how for these 3 here, that in the name, it's telling us origin and destination. So spinal cord, thalamic, thalamus. So it's going from the spinal cord up to the thalamus. That is representing an ascending pathway. So this has to be referring to a sensory tract. Similarly, spinocerebellar, spinal cord up to the cerebellum. That is ascending. So you need to pay close attention to the name. And then if you can also associate dorsal with sensory, right, that's going to allow you to identify ascending tracts versus descending tracts, sensory versus motor. So for these 4 major tracts, this table just kind of briefly refers to function here and also talks about what would happen if there were a lesion or damage to that particular tract. So each one carries very specific information, and we're going to look at those on the upcoming pages. With the dorsal column tracts, these are found in the dorsal region of the spinal cord, transmitting sensory information. So these axons transmit sensory information about conscious proprioception. So, our body sense that we are aware of. Like right now, I know that I am sitting down. I know that my knee is flexed. I know that my hands are resting on the table. I'm consciously aware of where my body is in space and how I want to manipulate it. Also, vibration and 2-point discriminative touch. So discriminative touch is very specific touch, right? Knowing like exactly where a sensation comes from on our body, knowing exactly where a mosquito lands and bites us. That kind of very specific type of touch is transmitted along these dorsal column tracts. All of the information is going to be conveyed to the primary somatosensory cortex so we can specifically localize where that sensation came from. With spinal thalamic tracts. So think spinal cord up to the thalamus. There's an anterior and a lateral spinal thalamic tract, and it's important to note the difference because they transmit different types of information. The anterior is going to contain axons that transmit pressure, texture, light touch. So not as specific touch, not that discriminative touch, but all the other properties of touch, which are very important. In contrast, the lateral tract transmits sensations of pain and temperature, right? So it's very distinguished here, anterior versus lateral. Same concept in terms of where it is going. It's going to go to the primary somatosensory cortex first, then association areas for interpretation. We're also going to see that with this sensory tract, that decussates or crosses over within the spinal cord. With spinocerebellar tracts, so going to the cerebellum, as we see here. So this has to do with motor coordination, right? If information is going to the cerebellum. So we have those proprioceptors in the muscles, tendons, joints, always giving feedback to the brain about our body's position. But with this type of information, it's more on a subconscious level. So body sense that we don't really think about, right? It's like that automatic sense of balance and positioning that our body does for us. That's the kind of processing that's going on here. So the cerebellum uses this information to coordinate muscle movements. With descending tracts, as mentioned, there's the upper motor neuron, there's the lower motor neuron, and the site of synapse here is going to occur within the spinal cord. So the lower motor neuron cell bodies originate within the ventral horn regions of the spinal cord and then extend as spinal nerves to innervate our skeletal muscle. So we have lateral, anterior, corticospinal tracts, reticulospinal, vestibulospinal. Notice how these all end in spinal, right? You got to think origin, destination. So corticospinal, cerebral cortex down to the spinal cord. Reticulospinal, think of a reticular activation system down to the spinal cord. Vestibulospinal, vestibular has to do with balance down to the spinal cord. So you have to pay really close attention to the name to differentiate sensory and motor. So similarly, the table here provides functions. We're going to look at each one on the upcoming pages. The corticospinal tracts or the pyramidal tracts are the largest in the cord, contain over 1 million pyramidal neurons and axons, and the upper motor neurons originate at the primary motor cortex. So here's our motor homunculus, here's our primary motor cortex being shown, right? That's where these neuron cell bodies are found. The axons then travel through the brain, travel down the brainstem, come to the medulla oblongata, and for the lateral corticospinal tracts, they cross over in the medulla. There is a smaller population of anterior corticospinal tracts that do not cross over, so we say that they are ipsilateral. So there is a smaller population which does not cross over. Regardless, when we focus on these green ones here, the lateral corticospinal tract, we see this crossing over event taking place in the medulla. These upper motor neurons then synapse with lower motor neurons, which extend out to innervate our skeletal muscle. So this tract, it gives off fibers or it synapses with lower motor neurons at all levels of the spinal cord. So we find that the tract gets smaller and smaller as it descends down the cord because of that extension that occurs. If there's any damage to this tract, there's going to be paresis, which is weakness in the muscle, or maybe even paralysis, the inability to activate and control the muscle. For that anterior corticospinal tract, that smaller population here, about 10% of the tract, does not cross over in the medulla. It's ipsilateral, meaning it stays on the same side of the cord. With this population here, it's going to innervate proximal muscles of the trunk, the neck, and it plays more of a role in influencing posture and gross movements, meaning more broad, like non-specific movements of the body, trunk, and neck. For the lateral corticospinal tract, that's innervating the bulk of our voluntary skeletal muscle. For the reticulospinal tract, this originates in the reticular formation area. And these upper motor neurons come down the spinal cord, synapse with lower motor neurons, and does innervate our skeletal muscles, not as much as we find with the corticospinal. And these play a specific role in controlling rhythmic gait patterns. So this is more control of our normal walking patterns and movement patterns that kind of automatically happen. And also, this tract here has fibers that control the autonomic nervous system. So, more subconscious control going on here. These stay ipsilateral, meaning they stay on the same side of the spinal cord. There's no crossing over that takes place here. And also is found that this tract plays a big role in inhibiting muscle tone and allowing for relaxation, which you may think like, why would you want to inhibit your muscles? But it's actually really important for control, ensuring that we have appropriate muscle activity, that our muscles aren't too stiff and spastic. So if there is damage to this tract, the muscle tone is excessive or hypertonic, meaning that the muscles are overly flexed, they're really rigid, it makes it more challenging to move the body and have normal activity. Then lastly, the vestibulospinal tract originates in the vestibular nuclei of the brainstem and travels down the cord to synapse with lower motor neurons. When we think about the vestibular nuclei, collection of neuron cell bodies, it's receiving input from structures that are involved with equilibrium or balance. So it receives input from the vestibular apparatus in the inner ear, receives information from the cerebellum, other motor areas in the brain, the basal nuclei. And processing goes on in that vestibular nucleus, and then that information is transmitted down the cord to play a role in controlling proximal limb muscles, help to really maintain balance and posture. So if someone has issues with this tract, issues with the vestibular nucleus, or then even issues with the inner ear or cerebellum, that will lead to ataxia, a loss of balance and difficulty with moving. So that's the end of chapter 12, the central nervous system, and we'll continue this conversation in chapter 13.